Bom dia, good morning, everyone. Welcome to our seminar today. So we have the pleasure to receive Dr. Julie Dunn. She's a research scientist from the National Institute of Health and Medical Research in CERN, which is a French public organization dedicated to biomedical and health research at the Cochin Institute of Paris. Dr. Julie, she co-leads a team for research that's named Functional Pharmacology and Pathophysiology of Membrane Receptors. So Dr. Julie is specialized in understanding membrane receptor and the molecular mechanism of action of these receptors, and she's particularly interested in the leptin receptor of NDPCRs. So she wants to understand the pathophysiology of obesity and type 2 diabetes. So very important research. And the primary targets of current available drugs in the markets. GPCR are of great interest. So welcome, Dr. Julie Dunn. And the title of her talk will be Get Receptive to Metabolic Receptors, the Key Players in Obesity and Diabetes. So thank you very much, Dr. Julie, for being here. And the stage is all yours. You can start. Thank you.
Thank you, Celia, for this introduction. And I would like to thank you also for having hosted us in your lab for this program, Capes Coffee Cube, and for having organized this seminar for us. And so last Wednesday, you heard Dr. Erika Secon presenting the biosensor that she developed in the team on Alzheimer's disease. And today, I would like to introduce you to metabolic receptors that we believe are key players in obesity and type 2 diabetes. So our team is located in the Institut Cochin in the heart of Paris. And you can also visit our team website if you go to the website of the Institut Cochin, if you want to know a little bit more about us. So why are we interested in membrane receptors? Well, these membrane receptors are at the interface between the extracellular environment and the intracellular part of the cell, so they signal specific information from outside to inside to lead to a specific biological response. And the deregulation of the signaling of these receptors can lead to the onset of pathologies such as obesity and type 2 diabetes. And so obesity is considered as an abnormal or excessive fat accumulation in the body that can impair health. So this is the definition of the WHO. And so there are multiple causes of the development of obesity, such as genetic factors or aging, but also there are environmental factors, such as lifestyle, inactivity and sedentarity, and healthy food, among others. So obesity is now considered as a global epidemic. And according to the World Health Organization, obesity is one of the most serious public health challenges of the 21st century. And indeed, if we look at the key numbers in 2022, we can see that almost half of the world population live with overweight and obesity. So this represents approximately 1 over 8 people in the world having obesity. And so these numbers just increases dramatically. So since around 30 years, obesity in adults increased by twofold, and mostly for children and adolescent obesity, the increase reaches fourfolds. So this is a really dramatic issue, and this obesity epidemic is not restricted to some specific countries, but our two countries, Brazil and France, are not spared, and the number is even higher in Brazil. So the issue of obesity is the fact that it has harmful consequences. And indeed, obesity is associated with the development of type 2 diabetes, liver diseases, heart diseases, or even give a higher risk to develop certain cancer. So I wanted to do a little bit of history about the obesity and to go back in time and to talk to you about leptin, which was the first hormone which was described in the field of obesity. So it was back in the 70s that Doug Coleman discovered that there was a circulating factor that accounted for the decrease of food intake and body weight, the regulation of body weight. So he used several mouse strains, so wild-type mice, and two laboratory mouse strains, which were the diabetic mice, the DBDB mice, and the obese mice, the OB mice. And so he just linked the blood circulation of those mice, and he found that while the DBDB mice was not really changed, he found that the OB mice, the obese mice, had a very dramatic decrease in food intake and a decrease in body weight, suggesting that the OB mice could lack a circulating factor and that the DB mice lack the receptor of this circulating factor. And it was only 20 years, more than 20 years later, that Jeffrey Friedman discovered the gene that encoded the OB gene that was sequenced by this researcher, and he found that this gene coded for a hormone that he called leptin, for leptos, meaning lean in Greek. And so leptin, now we know that leptin regulates food intake and body weight and also glucose homeostasis, but it's also a pleiotropic hormone that has multiple functions in reproduction, immunity, angiogenesis, and so on. So actually leptin is secreted by the adipose tissue, so it's an adipokine, and it must cross the blood-brain barrier in order to activate specific receptors in the hypothalamus. And this will lead to a decrease in food intake, an increase in energy expenditure, and this will then regulate in return the fat mass. So leptin will activate specifically some very specific neurons in the hypothalamus, the arcade nucleus hypothalamus, which are anorexigenic neurons, the POMC neurons, and it will inhibit the orexigenic neurons, the AGRP neurons, which are important in the regulation of food intake, and this will lead then to the control of energy balance. So the signaling of the leptin receptor is complex, but its main pathway is the STAT3 signaling pathways and also the PI3 kinase signaling pathways in order to regulate food intake and glucose homeostasis. So mutation in the gene of the leptin or in the gene of the leptin receptor can lead either in rodent or in human to severe obesity. And in patients which are deficient in leptin, the administration of exogenous leptin is able to rescue the phenotype with a decrease of body weight, hypoglycemia, and hyperphagia, and there's also a regulation of lipid metabolism. But the majority of the people with obesity do not have this genetic impairment, but rather they have a higher leptinemia, a higher leptin in the blood, to which they do not respond anymore. And this is called the phenomenon of leptin resistance. And there were several hypotheses to account for the leptin resistance in people with obesity. One of them was an impairment in the transport of leptin in the brain, or an alteration of the signaling of the leptin receptor. So in this presentation today, I would like to introduce you to this notion about the transport of leptin, and also the importance of having a good pool, a correct pool of leptin receptor at the cell surface. So the first question that we asked was, how does leptin penetrate into the brain and can reach the hypothalamic neurons? So in the hypothalamus, there is a specific region which is called the median eminence, and this median eminence is a circumventricular organ. It does not have the characteristic, the classical characteristic of blood-brain barrier with endothelial cells, but it contains specific specialized cells that we call tanocytes. And in collaboration with the team of Vincent Prévost in France, we showed that when fluorescent leptin is injected in periphery, the tanocytes then are able to capture leptin at the end feet of these cells, and will shuttle the leptin into the cell body of tanocytes to be released in the third ventricle in the cerebral spinal fluid. And actually, this work with active, competent leptin, but if we inject mutated leptin, which is still able to bind the leptin receptor but not able to activate it, we do not see any transport of this mutated leptin into the brain. And suggesting that the leptin receptor could be involved in this phenomenon. And indeed, later on, we showed that the complex formed between the leptin receptor and the EGF receptor is important to regulate the transport of leptin into the hypothalamus. And so we showed by many means that two receptors were interacting by immunoprecipitation, but also by using a specific methodology, technology, which is called time resolved FRET. So in this configuration, so the EGF receptor is expressed in the cells, and we can label the EGF receptor with a donor of energy. And when the cells are treated with leptin, which is labeled with the acceptor of energy, since leptin does not bind the EGF receptor, we do not detect any TR-FRET signal. It is only when we co-express in the cells the EGF receptor with the leptin receptor that the leptin, the fluorescent leptin, will bind to the leptin receptor and will allow the emission of the TR-FRET signal that you can see here in green. And we also observe that when this complex is activated with EGF, we have an increase in the TR-FRET signal, suggesting a modification of a conformation inside this ternary complex formed with the two receptors and the two ligands. And on tissue, so in the median eminence, we also observe the proximity between the leptin receptor and the EGF receptor using the technique proximity ligation assay. That is, it is detected in the wild-type mice, but not in mice which are deficient in the leptin receptor inside the tanocytes. And so why this complex is so important? Well, it's because EGFR is known as being a very strong inducer of ERK signaling. And in fact, ERK signaling, we showed that ERK signaling is very important in this process of regulating the transport of leptin inside the tanocytes. Inhibiting ERK signaling will inhibit the release of leptin from the tanocytes. And in the complex, the co-activation of these receptors will lead to a potentiation and increase in ERK signaling, which is necessary to release leptin in the environment, in the medium. So if we have an alteration of the transport of leptin in tenocytes by deleting the leptin receptor in these tenocytes, we would observe an increase of food intake in the mouse model. We will also observe an increase in fat mass and an alteration of the pancreas function with a decrease of insulin secretion upon glucose response, and also an intolerance to glucose when we inject glucose into the animals. So this example will show you the importance of assessing a hormonal access into the brain. And so leptin is not the only hormone that penetrates inside the arcade nucleus through this specific pathway. So now I would like to introduce you to another concept that we think to be quite important, is the pull of the cell surface receptor at the plasma membrane. And this is an important issue, particularly for the leptin receptor, for which we know that there are only 5 to 10% of the receptor which is localized at the plasma membrane. So we know actually that the leptin receptor gene could encode for two different proteins. One is the leptin receptor, it's a large protein, and the other one is a protein which is called endospanin 1. It's a very small protein. And we suspected that since these two proteins were linked by the same gene, that these two proteins could interact. And one of the particularity of endospanin 1 is the fact that this protein is only localized in intracellular compartment, and we observe it mainly in the trans-Golgi and the late endosomes. So we detected interaction between the leptin receptor and endospanin 1 that we showed in co-immunoprecipitation experiment, but also by using a specific methodology based on bioluminescent resonance energy transfer. So in this context, if two proteins are fused, one to the donor of energy and the other one to the acceptor of energy, and they are expressed in the cells and they do not interact, well, we will see this very linear signal of breadth, which is correlated to no interaction. But when the two proteins interact, like in the case of the leptin receptor and endospanin 1, we will see this saturation curve of a breadth signal, suggesting the interaction between these two proteins. And actually, what we observed was that the fact that endospanin 1 is retaining the leptin receptor inside the cells. By increasing progressively the expression of endospanin 1 on the left side, you see that the surface level of the leptin receptor is decreasing, while by silencing endospanin 1 in cells, we see an increase in the level of the leptin receptor at the cell surface. And this is also specific to the leptin receptor because we tested this effect for other receptors and they were not affected. And so by silencing endospanin 1, we have lower surface receptor at the plasma membrane, and by silencing endospanin 1, on the contrary, we have more receptors at the cell surface. So leptin will be able to activate better the signaling pathways involving STAT3, and this is what we observe. We have a higher phosphorylation of STAT3 when we silence endospanin 1. And one interesting point that we observed was that in obese mice fed with a high-fat diet, we observe an increase in the expression of endospannin 1 in the hypothalamus, specifically in the arcade nucleus of the hypothalamus. And so we were wondering whether decreasing the level of endospannin 1 in obese mice will have an impact on the development of obesity. So what we've done was to inject stereostatically, by stereostatic injection, we introduce an antivirus expressing shRNA against endospannin 1, and we then put these obese mice on high-fat diet or switch them to a normal diet. And what we observed was that wild-type mice have an increase in body weight, but endospannin 1, which were but mice which were silenced for endospannin 1 in the arcade nucleus and were fed with a high-fat diet, increased their body weight, but a little bit lower than the wild-type mice. But if we switch those mice to a normal diet, we observe that there is a decreased body weight, and associated with endospannin 1 silencing, this decreased body weight is even more important and sustained with time. And this decrease in body weight was associated with a decreased fat mass and also a decreased food intake. And the decrease in food intake was associated with a better capacity of leptin to signal into the hypothalamus, as you can see here, where in the condition of endospannin 1 silencing, you can see that leptin is able to activate better STAT3 phosphorylation. And later on, several groups also showed that the level of expression of the leptin receptor at the cell surface is very important in obesity, in other kind of model, the Bardet-Biedl syndrome or the Prader-Willi syndrome, for example. So this study showed that the correct level of a receptor at the plasma membrane can affect dramatically the regulation of a specific function, and in this case was of obesity and glucose homeostasis. And I would like to also continue with this aspect of regulation of surface pool of receptors by talking about CD36. So we know that endospannin 1 interacts with the leptin receptor in the arcade nucleus. But we also discovered that endospannin 1 interacts with CD36, which is a lipid transporter in the adipose tissue. And this has also a strong impact on the development of obesity. So we see that endospannin 1 is expressed in the adipose tissue in the mRNA and protein level, and actually it is the main adipose tissue in which this endospannin 1 is observed to be expressed. We detected interaction between endospannin 1 and the lipid transporter CD36 in different adipose tissue depots. And in adipocytes, we observe a colocalization between these two proteins in intracellular compartment. And in fact, the interaction between endospannin 1 and CD36 also impact on the level of CD36 at the surface of adipocytes. So this is observed when we bioetinylated the surface receptors, we see more CD36 at the cell surface in knockout adipocytes. And we also observe that there is more labeling of CD36 in adipocytes which are deficient of endospannin 1. And in fact, CD36 is an important transporter of lipids, so it will uptake lipid into adipocytes. So having more CD36 at the surface of adipocytes will accumulate more lipid in these cells. And this has an important impact because Um, when we compare obese mice, endospanning 1 and wild-type mice, which were challenged with a high-fat diet, we observe that these two mice have the same body weight and the same fat mass. But if we looked a little bit better into the accumulation of fat, we observe that the fat is preferentially accumulated in the adipose tissue instead of the liver. And so this decreases the steatosis of the liver, leading to a better inflammatory status of these mice. And indeed, endospanning 1 knockout mice have a lower inflammation that we observe with specific inflammatory markers in the blood or in the adipose tissue and in the liver. And because of all this, the endospanning 1 knockout mice fed a high-fat diet have also a better glucose tolerance. And all this observation that we've done in a preclinical model in mice was also observed, could be correlated with human data. And indeed, in human with obesity, we observe that lower insulin resistance is observed for patients with obesity that have a lower endospanning 1 expression in the adipose tissue. So this example, again, between endospanning 1 and CD36 reveal the importance of correlating of knowing the level of the receptor at the plasma membrane, and also reveal that a preferential accumulation of lipids in the adipose tissue can limit unhealthy obesity. And finally, so this is my last example to show you the importance of the pool of receptors at the cell surface. And this example concerns a very, very important drug target, which is the GLP-1 receptor in obesity and type 2 diabetes. So GLP-1 receptor is a G-protein coupled receptor. So it has 7 transmembrane domain and it coupled to G-protein and in order to signal. And so this receptor is responding to a hormone which is called GLP-1. GLP-1 is secreted by the intestine upon the meal, and it will act at several levels, but mostly in the pancreas to regulate insulin secretion when there is a high rise of blood glucose, and it also regulates food intake in the brain to decrease food intake. So there are several GLP-1 receptor agonists which are now medicines that have just revolutionized the treatment of obesity. And we performed recently work with this GLP-1 receptor at the level of defining the function of specific human variant of this receptor. So what we had done is, in collaboration with the team of Amélie Bonnefond in France, we have identified specific synonymous variants, non-synonymous variants in the exon of the GLP-1 receptor, and we try to detect with prediction the impact of this mutation on the receptor, and we selected the one which were predicted to be impactful, and we selected then 60 variants that you can see here, which are in red in the GLP-1 receptor sequence. And actually, we then study those mutations to better understand their impact on the function of the receptor. So we assess whether they have an impact on the expression of the receptor, on their surface localization, or on their signaling pathways. And one thing that we observed is the fact that many of them have a very strong impact on the trafficking of the receptor to the cell surface. And indeed, here in this panel, you can see that the level of the wild-type receptor at the cell surface is defined here at 100%, but the majority of these variants have a lower cell surface expression than the wild-type receptor. And 1/3 of this variant have a even lower level of expression by 50%. And because of this, many of these variants have a defect in their signaling pathways. And when we associated and we try to correlate the impact of this variant with the risk of developing obesity and type 2 diabetes, we observe a strong correlation. And so this human variant have a specific impact because they have impaired function, and so they won't be able to respond to specific drug which are used nowadays against obesity or type 2 diabetes. So this will be the next generation, the following study will be to identify indeed a specific drug that could help this variant to function and to help to build this personalized medicine. So the take-home messages of this talk would be the importance of membrane receptors in regulating energy and glucose homeostasis. I hope that I convince you that membrane receptors are important. The importance of hormonal transport into the hypothalamus to regulate energy and glucose homeostasis. That favoring the lipid uptake in the adipose tissue is also an important issue to minimize unhealthy obesity. And for sure, what is quite often neglected and overlooked is the fact that receptors at the cell surface are very important, and we can already have shown this with the leptin receptor, the CD36, and the GLP-1 receptor. So I will finish with this by just telling you to save your a specific week in March in 2025 because we are organizing with Professor Silvia Garcia and our team congress and a workshop on GPCR signaling and drug discovery. And if you want to learn a little bit more about this specific technology, BRET and TRFRET, this will be the occasion. This will be part of a discipline for which students can get credit, and this congress and workshop will be in USP. So I would like to thank all the people that were involved in all this work. So the team that you can see here, the different team members, and you can have also a look to the spaces. Maybe you recognize Erika Secon and also Ralph Jokers, which is the PI of this team. And you have also different old members that worked on this different project, and many of our collaborators, without who all this would have not been possible. Thank you for your attention, and I will be very happy to respond to your question.
Thank you so much, Dr. Julia Dem. This is an amazing talk, very interesting results. We open now for questions for the audience. People are thinking. So maybe I can say something. Sem perguntas ainda. So, well, thanks for advertising the workshop and the Congress. It's going to be a lot of fun also because you have a practical course, not only talks. And so this will be very interesting. So, so the potential, now there is so maybe now my later my question later. So the mutations that impair the trafficking of the receptor to the membrane are related to a specific region of the protein, like N or C terminal.
Not necessarily, actually. We try to define a region considering all the mutation, define a region that could be responsible for this, but we couldn't. They are spread in the whole sequence of the receptor. But what we know, what we think, is the fact that this mutation can affect also the stability of the receptor, so the conformation of the receptor is affected, and this conformation is not adapted to traffic actually the receptor to the plasma membrane, and so they will this receptor which are not stable, they will be degraded. And there are it's not the only example. The GLP-1 receptor is not the first example of proteins that have this defect of trafficking to the plasma membrane. You have, for example, the CFTR, which is a channel um, which actually also is not trafficking to the plasma membrane, but there is mean to change this, and the mean is to use pharmacological chaperone. So these are molecules that would bind specifically to the protein, to the receptor or to the channel, will stabilize it, and will then allow the quality control of the cells to say, okay, this receptor has a good conformation, it can go to the plasma membrane. And this, actually, this specific molecular chaperone is now used as a drug in cystic fibrosis, targeting this CFTR. So we don't know exactly which mutation will affect the conformation of the receptor. This cannot be predicted, and it does not necessarily affect a specific region of the receptor. Yeah.
Milena also has a question. Does leptin has an impact on areas other than the hypothalamus?
Yes, leptin is not only expressed in well, actually, the leptin receptor, because leptin is well, the leptin receptor is not only expressed in the hypothalamus, it's also expressed in other cell types, such as immune cells, because actually the leptin receptor is a type 1 cytokine receptor. So leptin is actually a cytokine. So it's also expressed in
Mm.
immune cells, yes, for example, and in some other cells involved in cancer.
Milena has another question. Production of leptin by a specific tissue.
Well, leptin is produced by the adipose tissue, and I'm not sure that I understand completely the question. Whether you mean adipocytes not located in the adipose tissue but somewhere else, do they produce leptin? I will say yes. If you have adipocytes that are accumulating in other maybe in other in you have also adipocytes in the breast. Those can also produce leptin. I'm not sure whether this was exactly your question.
Julie, the potential therapeutic strategy are being explored to enhance leptin receptor sensitivity in individuals with obesity related to insulin resistance in diabetes. So to increase this sensitivity of the leptin, are there potential any strategy currently?
Um, I think if we can target specifically the arcade nucleus, that would be a good strategy, yes. Because with the example that we have with endospanning, we indeed we can see that modulating the expression of this protein can affect the capacity of the leptin receptor to signal, to increase its capacity to signal in the arcade nucleus. Even with the low, because
Level.
yeah, we don't increase like 10 times. And it's a
Mm-hmm.
very subtle increase, but despite this subtle increase by 2 times, we are able to have an effect physiologically. So if we are able to do that specifically in the arcade nucleus, it would be a good option. But as one of the questions that the audience was asking is indeed the leptin receptor is not only expressed in the arcade nucleus, it's also expressed in other parts, I mean other cells, and immune cells can be a problem if you act because the
Mm-hmm.
leptin can is still, since it is a cytokine, it can affect immunity and it can
Mm-hmm.
increase inflammation in some specific cells.
Ah,
So if we
it's
can
very
target
tricky.
specific
Mm-hmm.
tissue, a good option.
That would be the option.
Mm.
It's becoming an epidemic, no? It's a real problem. The obesity and the diabetes.
Yes, and
It's,
it has been a journey for a long time by big pharma to find a way to find a medicines against obesity because before it was
mm-hmm.
not so efficient. There were many compound drugs that were released and after withdraw from the market because of side effect. And nowadays, with the GLP-1 receptor agonist and the receptor which are affiliated to this incretin GPCR, are a real promising field to have a very efficient drug against obesity. But still, I would like to warn we need to use this medicine with cautions because I know
Mm-hmm.
that there's a lot of black market also
Mm-hmm.
about this specific
Yes.
agonist. People try to get this
Mm-hmm.
agonist without consulting physicians because we need to be followed and it's only the beginning of it and we don't know the long-term side effects. But yes, there's since because of this epidemic of obesity, now we have a drug that can
Mm-hmm.
fight against it, but still we need to be cautious.
Yes. One more question here. Was any undesired phenotype observed in the animal model of ablation of anospanine? One undesired.
That's an interest. Yeah, that's a good question because I didn't go into details in the presentation. But when we looked at the knockout, the general knockout of this endospanning one, what we observe is that if we put the mice on high-fat diet, they become obese, but we have a better metabolically status with lower steatosis and lower glucose intolerance, low inflammation. So that was in the general knockout. But when we silence specifically in the arcade nucleus, since in the arcade nucleus it was only related to the leptin receptor, and in the regulation was a little bit complex for the leptin receptor, is the fact that we had two different mechanisms. One was to increase the leptin receptor at the cell surface. So in that case, we had a better STAT3 activation. But on the other hand, we had a decrease in PI3 kinase signaling. And it was because this protein was also involved in the activation of the receptor at the level of the PI3 kinase signaling pathways. And because of that, increased STAT3, decreased PI3 kinase, we observe regulation of decrease of obesity, but also an increase in glucose intolerance. So it was also a dissociation between obesity and type 2 diabetes. So that's why endospanin 1 was a little bit tricky molecule, protein to targets, because it has several functions at the level of the leptin receptor. It was not only at the regulating STAT3 signaling, but it was also decreasing PI3 kinase signaling. So that was the undesired phenotype that we observed in this case. Thank you for the question.
Very interesting. Fim das questões. Okay, so if we have no other questions, would you like to thanks again for teaching us so much, Dr. Julia Then. And we are looking forward to our workshop on March.
Yes,
On
save the dates.
2025, save the dates, and
students.
And ask the students to communicate to their friends and colleagues about these very nice opportunities for getting the credits and also for learning new methodologies for studying different problems in health or in other questions. Ok? So, muito boa tarde e bom almoço para todos vocês. Até uma próxima. Tchau, tchau.
Bye bye, thank you.